This is part 2 of our journey into biofunctionalization. With the base now clean and prepared, it's time to see how surface tweaks can help materials speak the language of biology. Here, you see 3 key parts: the base material, a chemical linker, and a biomolecule on top. But in real-world medical devices, we often stop at just the base and linker. That's because chemical functionalization alone can already make a surface more compatible, reduce fouling, or improve performance, without adding the complexity of biological molecules. However, in cases where we need biological specificity, like encouraging bone growth, guiding cell behavior, or sensing biomarkers, adding biocomponents such as peptides or antibodies becomes essential. While this approach is more complex, it's increasingly used in advanced implants and smart diagnostics, where tailored biological interactions offer clear clinical benefits. In nanomedicine, it's important to understand how molecules interact with the material, whether on the surface or deep inside. Absorption is when molecules move into the bulk of a material, like water soaking into a sponge. It's useful for drug loading or swelling effects, but not for precise surface control. Adsorption, on the other hand, is all about surface interaction. In physisorption, molecules stick using weak, reversible forces, like ionic attraction between positive and negative charges, or hydrophobic clustering, where water-fearing molecules group together like oil droplets. There's also hydrogen bonding, which is more like a light Velcro, just enough to hold things temporarily. For stronger, more permanent attachment, we use chemisorption, where atoms form covalent bonds by sharing electrons, like a firm handshake. A great example is a self-assembled monolayer, or SAM. It forms a stable, ordered layer through chemisorption and is often used to create reliable surfaces in implants, biosensors, or targeted drug delivery. Let's take a quick look at common chemical functional groups. These groups can be found in the biomolecules we want to attach, or at either end of a chemical linker, the head group or the terminal group. When preparing a surface for functionalization, our first step is choosing the head group of the linker. This part of the molecule needs to match the base material, whether it's metal, ceramic, or polymer, so it can attach securely. Meanwhile, the terminal group at the other end will connect to your biomolecule of interest. Think of it like a handshake. The head group grabs the material surface. The terminal group grabs the biological payload. The success of this anchoring depends on picking the right chemistry for each side. Different materials need different anchoring strategies. Titanium uses phosphate or catechol groups that bind to its oxide layer, while stainless steel needs cleaning before applying silane or polydopamine. Glassy surfaces like silicon or ITO, and ceramics like hydroxyapatite, alumina, zirconia are usually treated with acid or plasma to expose hydroxyl groups, then functionalized with silane or phosphate-based linkers. Inert polymers like PTFE or PEEK require harsher activation, while bioactive ones like PLA can use NHS or click chemistry. Gold and silver, as noble metals, form strong bonds with thiols, perfect for biosensing. The key takeaway: activate the surface first, then match the linker to the material for a strong, stable bond. Once the head group is anchored to the material, the next step is choosing the terminal group, the part that connects to your biomolecule, or in our analogy, the balloon. It must be reactive and compatible with your payload, like a protein, peptide, or drug, to keep it functional and firmly attached. First, let's look at the biomolecule you want to attach. For example, if you're developing a biosensor, you might want to immobilize an antibody onto the device. To do that, you need to understand the antibody's structure, especially its chemical functional groups, so you can target the right sites for binding. Here's a generic antibody structure. Common functional groups include thiol or sulfhydryl groups, SH, amine groups, NH2, carboxyl groups, COOH, and carbohydrate regions that contain aldehyde groups, CHO. Another example is if you're attaching a chemotherapy drug like cisplatin to nanoparticles for targeted cancer delivery. In this case, you need to know how cisplatin binds. It has two stable amina, NH subscript 3, ligands, and 2 label chloride ions. Lastly, if you're working with smaller probes or sensors, like DNA hairpins, commonly used in diagnostics or live cell imaging, you'll encounter phosphate groups, PO4 to the power of 3 minus, amine groups, NH2, carboxyl groups, COOH, and often terminal hydroxyl groups, OH, at the 3' or 5' ends. These molecules can also form hydrogen bonds, which are weak interactions involving partially charged atoms like oxygen, nitrogen, and hydrogen. Now that you know what functional groups are in your target, biomolecule, it's time to choose the right terminal end of the linker, and the reaction that'll tether this floating biomolecule balloon securely to the linker thread. This table shows common terminal groups used in biofunctionalization linkers and how they react with functional groups found on your biomolecule of interest. On the left side, from NHS ester down to vinyl sulfonate, are the reactive terminal groups of the linker. Across the top row, from amine to alkyne, are the common functional groups on biomolecules like proteins, peptides, nucleic acids, or small molecules. Each filled box indicates a recommended buffer and pH range that supports a successful covalent reaction. What's especially helpful is the color coding, which highlights both the type of functional group and the exact reactive site involved in the chemical bonding. For instance, red indicates carbonyl-based groups, orange for thiol reactive groups, green for imine structures, deep blue for alkene and alkane groups, light blue for haloalkanes, and so on. This mapping helps you quickly match the right linker to the available functional groups on your biomolecule. For example, look at NHS esters on the left. They react well with amine groups from the top row, especially in PBS or HEPAZ buffer around pH 7.5. The actual reaction occurs when the carbonyl carbon of the NHS ester is attacked by the amine, forming a stable covalent bond. So this table not only shows you what reacts with what, but also how and under which conditions the chemistry works best. This table works just like the previous one, with reactive terminal groups of the linker listed on the left and the functional groups on the biomolecule across the top. The filled boxes again show suitable buffer and pH conditions for efficient covalent attachment. Take click chemistry, for example. Either an azide or an alkyne group must be present at the terminal end of the linker, and the complementary group, alkyne or azide, must be available on the biomolecule. Under copper catalysis, they form a stable triazole linkage. This reaction is highly specific and bioorthogonal, meaning it doesn't interfere with other biological functionalities. This slide compares direct conjugation without a linker and conjugation that uses a linker. Without a linker, the biomolecule is attached directly to the base material, like glass, agarose beads, or membranes, using covalent chemistry. This works for simpler systems, but it limits flexibility. When a linker is introduced, especially in more complex or modular designs, it gives structural flexibility and can improve the biomolecule's function, such as solubility or targeting. Linkers can be cleavable or non-cleavable. Cleavable linkers are designed to break under specific conditions, for example, inside cells with high glutathione, low pH, or active enzymes, to release the attached molecule. This is useful for controlled drug delivery. Non-cleavable linkers, on the other hand, are used when the entire structure needs to stay intact, like in bivalent therapeutic systems. These systems, such as PROTACs, use a linker to connect two functional ends. One binds to the target protein, and the other recruits the degradation machinery. The linker holds the two parts together so they work as a unit. If the linker breaks, the function is lost. So choosing the right type of linker, cleavable or not, depends on how you want the system to behave in the body or in your application. In surface functionalization, sometimes we don't want the surface to be reactive everywhere. To prevent unwanted binding or background noise, we use inert groups to passivate the surface, like methyl groups, CH, subscript 3, or trifluoromethyl groups, CF3, which are hydrophobic and chemically stable. Another common choice is polyethylene glycol, or PEG chains made of repeating ethylene oxide units. PEG is hydrophilic, and instead of blocking through hydrophobicity, it creates a steric barrier that resists protein or cell attachment. If you're working with self-assembled monolayers, SAMs, or Silane chemistry, a common strategy is to mix reactive groups such as carboxyl, COOH, or amine open parenthesis, NH, subscript 2 close parenthesis, with inert groups like CH, subscript 3, or PEG. This lets us fine-tune the reactivity and spatial presentation on the surface. And when we want even more control, for example, to pattern reactive and non-reactive regions, we can integrate microfabrication or nanopatterning techniques, like the ones we've discussed in a video module earlier. This enables precise spatial control down to the micron or nanometer scale. It's quiz time. Stuck? No worries. Pause the video, check back on the previous slides, or feel free to Google it. In this module, we explored biofunctionalization, activating surfaces that allow specific interactions with cells or biomolecules. Step 1 is choosing a suitable base material, like metals, ceramics, or polymers, depending on the application. Step 2 is cleaning and activating the surface using dry or wet methods to expose reactive groups. Step 3 involves adding linkers that connect the surface to the biomolecule. And Step 4 is the actual conjugation, using covalent or non-covalent strategies while preserving function, often supported by inert groups or micro-nanopatterning.